All right, Christina, do you want to go ahead and let everybody in? Can you hear me? Yeah. Yeah. Thanks for your time. Yeah. All right, Sophia, are we ready?
Good to go.
All right, excellent. Thank you.
Good morning, everyone. Sorry about the delay. Thank you for coming to Natasha's defense. Rattled from having to drive across this town, but I'm thrilled to introduce her for her master's thesis defense. So Natasha came from Collins University, where she got her bachelor's in biology. And while she was there, she did an honors thesis on Acropora palmata populations over on St. John with Dr. Renee Goddard. And what's really remarkable about that is A, she published her honors thesis in Caribbean naturalists. And B, after working on Acropora palmata in the shallow zone, getting tossed around and making observations, she continued to want to work on Acropora palmata as part of her master's project. So I actually invited her to be a research assistant on our Acropora monitoring program. But then even more remarkable, while she's been at UVI, she helped to write and apply for a Water Resources Research Institute grant that she received. And Dr. Amy April, one of her committee members, is also on that project. And that is what funded her actual research assistantship and her project, which she's going to talk about today, which is using a multi-modal approach to assess Acropora palmata and disease around the northern U.S. Virgin Islands. She was also a Strong Coast Fellow. So one of her other committee members is Dr. Maya Trotz, who's the PI of that program. So she's also online. And with that, I mean, there's tons of other things to talk about Natasha, because she's also known as Mama Tosh. She takes care of us in the field. She's just been an incredible person to have in the lab. And with all that advertising, you may want to hire her away, but you can't because I'm hiring her to continue on to work with us.
All right. Thank you so much, Marilyn. And thank you everybody for being here for my thesis defense titled Using a Multimodal Approach to Assess Acropora palmata Populations and Disease in the Northern U.S. Virgin Islands. Coral reefs are vital ecosystems, and while they only cover 0.1% of the ocean, they are relied upon by over 500 million people for food, protection, and income. Additionally, they are home to the greatest number of species, only second to the rainforest, including over 800 species of coral and over 4,000 species of fish. One of those species, and my focal species, is Acropora palmata, or commonly known as elkhorn coral. This species is a relatively fast-growing species and is easily recognizable due to its branching morphology. This species is found throughout the wider Caribbean, and its geographic range can be seen here on the map in yellow. This species is very important due to its large branches, and it provides critical habitat for many ecologically important species like fish and invertebrates. Additionally, this species is very important to coastal communities as they protect coastlines by reducing wave energy. Unfortunately, this species has been severely impacted by disease, bleaching, hurricanes, and predation, causing its population to decline by approximately 97% in the past 40 years. This steep decline resulted in the species being listed as threatened on the Endangered Species Act in 2002, listed as endangered on the IUCN Red List in 2008, and relisted as critically endangered on the IUCN Red List in 2022. One of the two primary diseases responsible for the decline of the species is whitepox disease. This disease can be identified by these white patchy areas of tissue loss. This disease was identified in the Florida Keys in the mid-1990s, and then in the early 2000s, the causal agent Serratia marcescens, which is a fecal bacterium. A later study linked this bacterium to sewage contamination and reef water around the Florida Keys. Unfortunately, this link between Serratia marcescens and whitepox disease has not consistently been found since its identification. A study in 2015 found that only 26% of whitepox lesions were positive for Serratia marcescens. And this trend of inconsistent identification of whitepox lesions associated with Serratia marcescens is also found here in the US Virgin Islands. Several studies, including one of our own master students' thesis research, found that whitepox disease is not always associated with the Serratia marcescens pathogen. Additionally, these studies have found lesions containing other known coral disease-associated bacteria, like Vibrio and Eranthemonas. Because of this inconsistent connection with the identified pathogen Serratia marcescens, you have to wonder if there's just one pathogen causing these lesions. So the original rule of thumb is that there's one pathogen for one disease, but we've come to realize that that's not necessarily always the case. More than likely, it's the fact that corals only have so many ways of displaying disease, and that there's a whole host of different factors that are involved in this. Now, looking at diseases, we realize that it's a very complex system that contains a whole bunch of different factors, from factors associated with the host, the environment, and the microbiome. So let's go ahead and take a look at these components in regards to whitepox disease through a more local lens. First, let's take a look at the host. Well, a prior study of whitepox disease in St. John between 2003 and 2009 determined that larger colonies were more likely to present with whitepox disease. And it was suggested that this was due to age and a weakening immune system, or that it could be due to surface area that's getting exposed to the pathogen in the water. This study also found that density of colonies did not seem to be playing a role in the disease prevalence. While not many other studies have looked at demographics in conjunction with whitepox disease in the USVI, we were lucky enough to have a demographic monitoring program between 2011 and 2013 that found that whitepox disease was the most prevalent disease affecting our Acropora palmata populations in the USVI. Additionally, this program has been reestablished and will allow us to continue to monitor Acropora palmata populations and gather more long-term data. Now, let's go ahead and take a look at the environment. While the US Virgin Islands have very steep topography, and due to our near surface bedrock layer, water retention in our soil is very low. So during rain events, we get a lot of stormwater runoff that leads to nutrient and bacteria inundation in our near shore reef environments. And this is most likely due to leaky septic systems and faulty sediment traps in areas of construction. To monitor near shore reef environments, the USVI does have 2 water quality monitoring programs currently. They have the ambient and the beach sampling. Unfortunately, these programs only sample surface waters in these areas and do not sample water at the depths of the coral reefs. Additionally, while studies around the USVI have collected sediment and seawater samples for microbial analyses, these have not necessarily been done in the Acropora palmata habitats. So last but not least, let's go ahead and look at the information we have on the microbiome. Studies have confirmed that microbial communities vary depending on the portion of the coral that you're examining, and each one of these plays a different role. Studies have found that these microbial communities can also be altered by the external environment. Currently, there are very few studies looking at these microbial communities in Acropora palmata, and even fewer in the US Virgin Islands. Thus, this study aimed to incorporate the elements of the host, the environment, and the microbial communities to examine whitepox disease through a more holistic lens, using disease demographics, water quality, and microbial assessments. Given that whitepox disease has been linked to fecal pollution in the nearshore reef environments, I hypothesized that the greater levels of fecal indicator bacteria would be associated with greater disease prevalence. For my second hypothesis, I predicted that during times of higher amounts of rainfall, like we see during our wet season, that we would have higher levels of fecal indicator bacteria in those nearshore reef waters. For my third hypothesis, given that several studies have seen differences in microbial communities of coral mucus, sediment, and seawater, I too hypothesized that we would see that in this study. And lastly, I hypothesized that microbial communities of coral mucus collected from the diseased colonies would be different from the coral mucus collected from healthy colonies. So let's take a look at how this was all done. My study took place in the US Virgin Islands, which is a territory just east of Puerto Rico. The US Virgin Islands are comprised of 3 different islands: St. Thomas and St. John to the north, and St. Croix to the south. While Acropora palmata is located around all 3 of these islands, for ease of access, I selected 7 sites around the northern 2 islands of St. Thomas and St. John. These sites are the location where those demographic monitoring program has been reestablished. There are 2 sites on the south side of St. Thomas, Cow and Calf and Flat Key. 3 sites on the north side of St. Thomas, Bonny Bay, Inner Brass, and Thatch Key. And then 2 sites on the south side of St. John, Reef Bay and Yazoo Point. At each one of these sites, I collected water for water quality assessments, performed surveys for demographics, and collected samples for coral, seawater, and sediment microbial analyses. So as I mentioned, at each one of these sites is a reestablished demographic monitoring program. And so each one of these sites has 3 circular plots, 7 meters in radius, that have a minimum of 10 to 12 tagged colonies, and a random number of untagged colonies as well. These plots were assessed starting at the start of the study. And from these assessments, I looked at the maximum dimension of the colony, the density of the plots, and I was able to go ahead and get an estimate of disease prevalence by looking at the percent of the tagged colonies that had white pox lesions. For the water quality, water samples were collected by hand approximately 1 meter above the reef substrate. These collections occurred at each one of these sites once a month for 3 months in the wet season of 2021 and 3 months in the dry season of 2022. The samples were analyzed for fecal indicator bacteria, specifically Enterococcus species and E. coli, along with total suspended solids, and these were analyzed at UVI's environmental analysis lab. I also looked at nutrients, specifically dissolved inorganic nitrogen, ammonium, total nitrogen, and these were sent to an outsourced lab back in the States. Microbial sampling, this occurred in late August of 2021, and I collected seawater, coral mucus, and sediment at each one of these sites. For the seawater, I collected 2 site-level seawater samples, and then I also collected seawater from directly above each one of the colonies that I sampled. For the coral mucus was collected by gently agitating the coral surface until mucus was visible and freely suspended. Then the mucus was collected using a 10 milliliter syringe. On diseased colonies, coral mucus was collected from both the diseased lesion margin as well as from the apparently healthy area of the colony that was at least 10 centimeters away from the diseased lesion. On healthy colonies, we also collected coral mucus, and the number of diseased and healthy colonies sampled did vary by site depending on whether or not disease was actually present in the demographic monitoring plots. Lastly, sediment was collected from the substrate at least within 1 meter of each one of the colonies. After collection, all samples were frozen and placed into a dry shipper, and then they made the journey with me from the USVI all the way up to Woods Hole Oceanographic Institution on Cape Cod. It was here that I was lucky enough to be able to work under and learn from Dr. Amy April and her amazing lab members. I also wasn't too sad about getting to wear some sweaters and hats again. So up in Woods Hole, I extracted DNA from each one of my samples and then targeted the V4 variable region of the 16S ribosomal RNA gene for sequencing. The samples were then sent to MIT where they were sequenced using an Illumina-MiSeq. After I received the sequence results from MIT, I identified each one of the bacteria down to the genus level and performed the analyses all using R. All right, so let's go ahead and dive into the results. First, let's take a look at the demographic results. This here is a plot of the average maximum colony dimension. On the x-axis, we have the sites, and on the y-axis, we have the average maximum dimension of each one of the colonies in centimeters. The letters denote, or the letters above the bars denote significance. And here we can see that Flat Key and Inner Brass had significantly smaller colonies than Reef Bay. And while not necessarily significantly different from the others, we do see that on average, Thatched Key did have the largest colonies. So given previous literature suggesting that as colony size increases, so does disease prevalence, we would expect to see the same patterns reflected in the disease prevalence estimates. So here we have a plot of the disease prevalence. Again, site is on the x-axis. And on the y-axis, we have the average disease prevalence in percent. While we do not necessarily see those same patterns reflected from the colony size here in the disease prevalence, we do see that Thatched Key had the largest colonies and also had the greatest disease prevalence. I do want to note that there was no disease seen at Botany Bay, Cow and Calf, and Inner Brass during these surveys. Here we are looking at a plot of colony density. So again, on the x-axis, we have site, and on the y-axis, we have the average density, or the number of colonies per meter squared. And what we see here is that density did not vary significantly among the sites. So if you'll remember back to my hypotheses, I hypothesized that fecal indicator bacteria would be positively associated with disease prevalence. Well, unfortunately, due to the timing of all my different sampling periods, I was unable to address this hypothesis, but no worries, let's move on. So now we're gonna go ahead and look at the water quality. First, let's go ahead and look at the fecal indicator bacteria and Enterococcus. On the x-axis, we have the season, and on the y-axis, we have the number of colony-forming units of bacteria per 100 milliliters. Now on, I'm just going to say CFUs, it's a lot easier. When I tested for the effective season, I found that Enterococcus species were significantly higher in the wet season, and that's exactly what I hypothesized. However, I do want to note that the local threshold for unsafe levels of Enterococcus is approximately 110 CFUs, so the values that we're seeing here are actually quite low. I also want to note that we did test for the effect of site as well as the interaction of site and season, and neither one of those were significant. So now let's look at the other fecal indicator bacteria, E. coli. So on the x-axis, again, we have season, and on the y-axis, we have the CFUs of E. coli. Here, while E. coli was higher in the dry season, there was no significant effect of season on the E. coli levels. While I'm not exactly sure why E. coli levels were higher in the dry season compared to the wet season, the literature does suggest that E. coli can persist in the environments in the sediment as well as on macroalgae. And so maybe what we were seeing here was a little bit of wave action increase in these environments. And so a little bit of stirring up of those sediments, and we were able to collect a little bit more of the E. coli. Although I didn't really test for that or look for that. So ultimately, I do have to reject my hypothesis for this fecal indicator bacteria. And now, while I did not hypothesize about the overall water quality, I would be remiss if I didn't actually explore those overall trends. So to do this, I created an index and I scored each reported value that I received for each parameter, being the fecal indicator bacteria and the nutrients, by their reliability on indicating fecal indicator or fecal sewage in the environment. So for example, if I got a reported value of 12 coliform units of Enterococcus, I would then bin it as medium and give it a score as 2. And then based off of the weight that I applied to each one of these parameters, again, based off of their reliability of indicating sewage in the environment. So for this one, it'd be weighted as a 3. So I'd multiply 2 by 3 to get 6. And I would do this for each one of the reported values for each one of these parameters. And then I would add them all up and have a total score for each one of the sites. And what this did is it ultimately allowed me to look at each one of these sites holistically instead of looking at each individual parameter. So then I looked at these across time. Here we have a map of all my study sites. Across the bottom, we have the color code for the total scores. And now we can see how water quality varies through time. This now allows us to have a baseline of overall water quality across all of these sites. So let's get to what I consider the juicy part of my thesis. Let's look at the microbial communities. So here is an MMES plot, and this allows us to visualize the similarities and dissimilarities of the microbial communities of each one of my samples. So each point represents a sample and its bacterial community. You'll want to focus on the distance in between each one of the points. The points that are further from one another have greater variability in the microbial communities. You'll also want to look for clustering of points because those indicate that the communities are fairly similar. On this plot, the sediment or the sample types are denoted in different colors. So mucus is in dark blue, the sediment is in orange, and the seawater samples are in the light blue. So let's go ahead and look at the sediment samples. Here we see that the majority of these samples are fairly close together and in a cluster. This means that the microbial communities of the sediment samples are fairly similar. However, we can see that they're far away from the points of the mucus and the seawater. And this means that they're probably quite dissimilar from both mucus and seawater samples. So now let's go ahead and take a look at the seawater. Again, we see that these points are very close together in a cluster, and this indicates that these microbial communities are fairly similar. They're also pretty close to the mucus samples on this plot, and so that means that they are probably similar to the mucus communities, but they're still different in a way. And now let's look at the mucus samples. So here we see a little bit of clustering up towards the top, but we also see that there are some points that are fairly far away from one another. And this indicates that some of these samples, their microbial communities are probably fairly similar, and others are highly variable. And through use of a Permanova, I found that indeed, these microbial communities were significantly different by sample type. But what's going on with the mucus samples? Let's go ahead and take a look. Here we have another NMPS plot. This time we're looking at just the mucus samples. So on this plot, the colors correspond to the areas of the colony that I collected the samples from. And then when we look at this, we see that that clustering that we saw on that previous plot is actually due to the clustering of the healthy samples. This includes the healthy samples or the healthy on the diseased colonies, but then also the healthy samples from the healthy colonies. And we see that those are fairly similar to one another. But when we look at the disease samples, we can see that those points are fairly spread out. And so this means that the diseased samples, their microbial communities are probably pretty variable. And they're also different from the healthy samples. And then through a Permanova, I confirmed that these diseased mucus microbial communities were significantly different from the healthy mucus microbial communities. But how exactly do these microbial communities differ? And is the pathogen Serratia marcescens that's associated with whitepox outbreaks in any of my samples? Well, the answer is no. We did not find any Serratia marcescens in any of my samples, but if there's not Serratia marcescens, what else could be going on? Well, in order to answer that, I created this bar plot of relative abundances of the top bacterial families seen in these samples. So on the x-axis, we have the samples, and on the y-axis, we have the percent relative abundance of each one of these bacterial families. Here is we see dysbiosis. We see a shift in the bacterial communities from healthy to diseased. We're seeing some families are decreasing in relative abundance, and we're seeing some families that are increasing in relative abundance. But what's more interesting is that two of these families that we see increasing in relative abundance in the diseased samples are known associated bacteria with coral diseases. So Rhodobacteriaceae and Viverinaceae. Rhodobacteriaceae has been found in association with stony coral tissue loss disease. And certain species of Viverinaceae were seen in other studies associated with whitepox lesions. And now, while I can't say that these are specifically causing the disease, we can start to see those puzzle pieces come together. So I hope at this point I haven't lost any of you, but if I have, I'm going to go ahead and summarize everything up. So my first hypothesis was that fecal indicator bacteria would be positively associated with whitepox. Well, again, I wasn't necessarily able to test that due to methodologies. My second hypothesis was that fecal indicator bacteria will increase during our wet season, and yes and no. And our coccus levels did, but that wasn't the same for E. coli. And then when we look at our third hypothesis of microbial communities varying between the different sample types, we did see that. We saw that those microbial communities were significantly different. And for my fourth hypothesis, looking at the microbial communities and whether or not they would significantly differ between the healthy and the diseased coral mucus, well, yes, again, they did. So what does this mean for the future? Well, water quality is constantly changing, and we can see that it was highly variable. So this data creates a really good baseline, and continued monitoring will help us determine trends and identify any abnormal changes. And luckily, that's what we're already doing. We're continuing this monitoring. So at least we'll be able to see what's happening in the future. After further investigation, Enterococcus species, as well as E. coli, can come from multiple different sources, and they're not necessarily always indicative of human fecal matter. However, there is a human fecal marker, HF183, that is currently being used in California, looking at whether or not fecal indicator bacteria are actually related to human sewage. So future studies around the USVI should look into this. While I did look at some of the key demographic parameters of our Acropora palmata populations, I did not look at their genotypes. And this information is a huge piece of the puzzle that needs to be addressed, and may potentially explain why we saw such high disease prevalence at Thatch Key. Because if all of those colonies have the same genotype, they're most likely less resistant to the disease than other colonies at other sites. And luckily, Dr. Eric Saka at the College of Charleston is already looking into this, so stay tuned. Very few studies have looked at microbial communities in Acropora palmata, and even fewer still have looked at them in the US Virgin Islands. The literature suggests that microbial communities are influenced by the environment, so this creates a really good baseline as our coral reef environments are changing. And so we'll be able to compare that in the future. And lastly, the comparison between diseased and healthy mucus microbial communities showed evidence of dysbiosis and identified two coral disease-associated bacterial families. But this is just the tip of the iceberg, as disease processes are super complex. So continued studies are needed to see if these patterns continue to persist. And at this point, I'll go ahead and thank my funding sources. Also thank my advisor, Dr. Marilyn Brandt. Thank you so much for having me in your lab. Thank you to my committee members, Dr. Amy April and Dr. Maya Trotz. Thank you for your guidance during this whole process. I do want to thank the grant lab members, specifically Lindsay Dade. Thank you so much for all of your help with water quality. And I don't think I would have stayed sane if it wasn't for you. I do want to thank Catherine, Sonora, Brad, Dan, and Jason. This sampling wouldn't have happened. Also, morale would have been really low if you guys weren't there. I do want to thank the April Lab. You all were so welcoming and were so willing to teach and let me ask questions. I do really want to thank Cynthia. Without her mentoring, I don't think I would have made it through all the data analysis that I had to do for the microbial communities. I want to thank my 2020 cohort. You guys were amazing. We went through COVID together and we're still here. I want to thank Amber, Rozzy, Cindy, Addie. I do need to thank my two professors from my undergrad, Renee Goddard and Morgan Wilson. Without them, I wouldn't be here because I wouldn't have been down in the US Virgin Islands and realized that I love marine biology so much. I have to thank Lainey Metz. She was super supportive during the writing process. And then last but definitely not least, I have to thank all of my family and my friends. They were vital to this whole thing going off without a hitch. Special thanks to Jenna Setzman, my sister Brianna Bestrom, and then my boyfriend Alex. They definitely were there to make me laugh when I wanted to cry, and they were there through all of the emotional turmoil that I was going through this entire process. So it wouldn't have been possible without them. And at this point, I will go ahead and take any questions that you might have. All right.
The start of this process. We had a great presentation. So now we're going to do questions from the audience, probably starting first in the room and then Zoom. And then the committee will have a closed session. So the committee members can either ask some questions publicly or just save them all.
And Sophia, would you mind kind of moderating some of the questions that are in the chat and on Zoom? I'll go ahead and ask the questions and see if anybody has questions in the room first, and then we'll move to Zoom.
Sounds good. Sure thing.
Awesome. Thank you. Is anybody in the room? Tyler?
The pattern you saw where the healthy tissue on diseased colonies
Yes.
was similar to the healthy tissue on undiseased colonies.
Yes.
And then, of course, different from the disease. What do you think that says about what's driving the disease?
Well, I think that could potentially be more so in terms of like what's in the water column and what's actually settling on top of the colonies. Because you could potentially have just a very localized area that has that, response to that. It could also be in relation to, just in terms of the immune response of the coral. So it may just be that isolated area had some issues, and considering that I sampled so far away from that lesion, that it hasn't quite progressed to those other polyps on the colony.
So, and do you think, because I know some people have brought up that apoptosis is a driver, that just it's the coral itself in some respects that's driving the tissue loss.
Right.
I mean, how would that, do you think the pattern you saw would indicate not apoptosis?
I honestly not quite sure. I think that if it was apoptosis, I think that we probably would see, I feel like a lot of the lesions that we saw were fairly all over the place on the colonies. And so, potentially that might be the case. But overall, the health of the rest of the colonies were the healthy part of the colonies were looking very healthy. Like they weren't looking like they were starting to bleach or having some, issues going on there. So, I would say that they're probably, just more localized.
You might also not repeat the questions.
Oh, yeah, thank you. Sorry, for the audience, Tyler asked, since I saw
Natasha, we could actually hear him pretty well.
Oh, perfect. Awesome. Thank you, Sophia.
Yeah.
Wonderful. Any other questions in the room? Allison.
Great presentation, Tasha.
Thanks.
Knocked it out of the park. One of the really interesting things that you had on some slides that you didn't get to talk about so much, and I know you had a lot of things, were the graphs where you have where the dots of the colors change depending on low and high amounts of different bacteria.
Yeah, the water
and
quality
dry
index.
seasons.
Yeah.
Yes, exactly. So can you please speak a little bit more about that? Because I think it's super interesting and, you know, in relation to specific sites, what do you think is driving those changes between the seasons? And then also, you have so much going on with your work, but what do you hope to do with that data? You know, obviously there's some great and not great things happening with the water quality. So how will you then use that to help, you know, the environment? Is it going to go to like managers? You know, what's happening next?
Yeah. So in terms of what might potentially be going on at those different sites, so, you know, certain sites were closer to high areas or high densities of anthropogenic impacts. And so some of that might be that because certain sites were closer to those areas, you're seeing, you know, potentially higher levels. And then some of that I think also has to do with hydrology. So depending on, you know, exactly where those sites were located, how much wave action you have in those sites. And then, you know, most of these sites are fairly close to land. They're like very near shore, but some of them are removed from like the main island, or at least removed from different places that have quite a lot of people that have developed in those areas. And then in terms of, you know, what I see, you know, going forward with this data, you know, it does vary quite a bit. And so really, truly, we do need like a long-term set of data to really make some like hard conclusions about like what's happening there. And so I think that this definitely needs to be shared with DPNR, just because, you know, they are already doing some of this water quality testing at other sites, but they're not necessarily doing them at these sites. And they're not doing them at the reef level. And so it is definitely something that they need to be aware of what's going on, just even to have it in just a database, so that they can go back and, you know, look at that if things continue to change. Yeah, of course. Rachel, I believe you had a question. Yeah.
So my question is kind of like looking at your data and whatnot, do you think this kind of shows that amputation could be a good intervention strategy when you do see white pox? So like, especially with palmatopopulations, they're easy to kind of cut the branches and stuff off. So like, if someone does have white pox, like, would it be beneficial to maybe just go ahead and amputate that and try and save the rest of the colony based off, you know, your healthy sample than your disease sample?
Yeah. Well, I mean, I will say that I wouldn't probably go to that, just because like some of these lesions are like dead center in the center of the colony. So that wouldn't necessarily be applicable in those instances. Additionally, when you're doing that, you're also stressing out the colony. And so are you really helping or hindering in that fact? Also in terms of, you know, what we've seen here, and I've actually gone back and looked at some of these colonies since they were the tag colonies and looked at them like a year and a half later after we've done some of these reassessments. And some of the lesions have actually completely re-healed. And so should we put them under that stress of just, you know, lopping off one of their branches? Just, I mean, I don't feel like it's really necessary at this point, unless we were seeing it to be like fairly like bad in those cases. But I really probably wouldn't go to that as a course of, you know, mitigation.
Going off the roots with the mitigation efforts, if it's seeing that the bacteria is similar to what's in certain core tissue loss with the rid of back rolls, could you treat with similar amoxicillin the same way you would?
I mean, potentially, I don't think that that's been looked at all. Yeah. Yeah, I don't think that that's been looked at all, but it's something that you could definitely look into in the future and see, yeah, does that actually like help speed up the process of recovery from these, you know, lesions? I don't know. No. All right. Any other questions in the audience? All right, Sophia, can we go ahead and start looking at the questions in Zoom?
Velma Tyson had a question. So Velma, if you still have that question, feel free to unmute. We also had lots of congratulatory praise in the chat. Amazing job, amazing presentation, amazing work. super. So thanks, Gwen, Mallory, Jenna, Cynthia, and all the others who posted those in the chat.
So this is Velma. Thank you. The question had already been answered, asked by someone else.
All right, great.
Thank
Thanks.
you.
Thank you.
All right, any other questions on Zoom? Hearing none, let's give Tasha one more round of applause. Great job.
We're gonna take like a 5-minute break and then we'll move into the closed.
Thank you, everybody. Thank you, everybody on Zoom.
Yeah, thanks for attending, everyone.
Lady, Lauren, Brittany.